At Amaryllis Nucleics, we've developed the most efficient method for producing RNA sequencing samples. So let's see how it works. We start with an RNA molecule, and we prime it with a small piece of DNA called a primer. We use another enzyme to make a copy of the RNA called cDNA. Now, here's where we start to get different. We take advantage of a process called terminal breathing. And what terminal breathing is when DNA, RNA, or cDNA in their double-stranded form open and close at their ends. And this is a function of thermal energy, even at low temperature. When the ends open up, this is when our adapter can interact in a process we call breath capture. We use a second enzyme to make this interaction permanent. And this alone saves several steps over competing methods. Following breath capture, we do a PCR multiplication to amplify the sample for sequencing. The entire process is just 4 steps and takes only 4 hours.